again. Maybe I've seen some of you in September. I gave an introductory talk to the Ensembl project and the resources that we had. We mainly focused on the genes and transcripts, so we were thinking about how Ensembl takes genome assemblies and annotates them with gene models, which is obviously formed of, each gene is formed of one or a number of different transcripts. We were thinking about how we can get different types of data for those genes and transcripts. So having spoken with Jenny previously, but before the webinar started, we did write down a few different important pieces of data and views within Ensembl that Jenny at least thinks that would be really useful for you guys to explore. So the main category that the rest of the data falls under is variation and phenotype data. So that's what we're going to be focusing on mainly today. It's going to be a little bit shorter than last time. So we're also going to look at a tool that we have in Ensembl called the VEP, the Variant Effect Predictor. And then we're going to have time for some questions at the end as well. I think Jenny has some that she sent to me, and I can answer those and answer any other questions from you guys as well. So as I mentioned in webinar 1, we were just mainly exploring Ensembl, the genes and transcripts. And then in this webinar today, we're going to look at the variation data that can sometimes be associated with different phenotypes and diseases. And then we're going to look at the VEP, which is short for the Variant Effect Predictor. I've already answered this question, but this is just to let you know that if you do have any questions that come up during the webinar, please just shout out. I can't really see the chat box, so please just do mention if you have a question, and I can answer them throughout the course of the webinar as well. This is just a quick slide to show you where the training materials are for this webinar. So I think some of you might be new and you didn't attend last time, but all of the course materials for both webinars are available on an online training resource that we have. So I'm just going to jump out of the presentation now quickly and show you where to find that. So if you go to if you open up a tab in your browser and type in training.ensembl.org and navigate to this website, you can see that we have information about our different training events. And in the bottom left-hand corner, there's a link where you can find training materials. So if you click there and scroll through our calendar to the correct date, we're actually going to be looking at the 14th of September, in fact, because this is when we had the first Biocurators webinar. So you need to go down to September for us, even though we're in October now. And here you'll be able to find information about the webinars, but you've got a presentation, PDF of the presentation from last time and this time, and then also a course book that covers all of the materials that we're going to cover, which is screenshots of the walkthrough demonstrations. So hopefully you can get those, access them, download them, and use them as you need. So they're completely free for you to use and disseminate to your lab members as you want to. Okay, so now we're going to start thinking about the variation data in Ensembl. So first we're going to think about how in Ensembl we classify variants. We're going to think about what species has variation data within Ensembl and where Ensembl gets that variation data from, so the sources and the databases that we retrieve the variation data from to display. Then we're going to think about accessing the variation data through the Gene tab and the Variation tab in Ensembl. And then we're going to look at this tool for analyzing and predicting the effects of variants. It's called the Variant Effect Predictor. So this might be quite basic, but I think it's quite an important idea to cover. First up is the idea of variants and how they can differ in terms of size. So the first is small-scale variants, and these, as we define in Ensembl, is anything that affects less than 50 base pairs. So these might be things including single nucleotide polymorphisms, so SNPs, or small insertions and deletions as well. So you can see, as examples of these, we have a deletion of a C in this blue individual, or the insertion of a G. So these would be examples of deletions or insertions. And then the single nucleotide polymorphisms is where there's no change in, there's a change in the sequence rather than a change in the number of bases. So this is rather a T to a G rather than an insertion or a deletion. The second type of variance is the large-scale variance. So this is anything that affects more than 50 base pairs, which is, again, it's an arbitrary definition that we have in Ensembl. And these can include things like copy number variants, large deletions, duplications, insertions, translocations as well. So these mainly affect larger chunks of chromosomal sequence. So when we display and think about variants and variation data in Ensembl, we like to think of them in terms of their consequences. So where a variant falls along the genome in relation to the genes that exist in that region can be really important for the effect and the consequence that a variant has. So if a variant falls, for example, outside a gene, it could fall in the upstream or the downstream region. This obviously has a less direct effect on the protein structure and function, but we do know obviously that there are regulatory regions that fall outside of genes, and so even variants that don't fall within the coding regions of a gene obviously do have the potential to affect transcriptions and obviously the expression of genes. Variants obviously also can fall within genes, so these might fall again within non-coding regions, so in the UTRs or in a splice site or an intronic region. And obviously these variants, again, they don't have a direct effect on the protein structure and function, but can still affect how the mRNA is processed after it's been transcribed, whether it's expressed, whether it's degraded, or whether it actually does produce a protein in the end. So again, these variants have an effect on protein expression in the cell. We have two types of variants that fall within the coding region. So these can be either synonymous variants, and these are variants that do not change the protein sequence. So obviously we know that the triplet codon, the triplet code of the genome is degenerate. So more than one triplet codon can encode a single amino acid. So a base pair might actually still encode the same amino acid, and in this case we call them synonymous variants. However, variants can obviously also change the protein that's encoded as well, the single amino acid. So in this case, we call them non-synonymous or missense variants. So these are variants that have changed the amino acid that's encoded at that particular position. So what we do then in Ensembl is we annotate each of our variants with a consequence term. And these are based on sequence ontology, so SO terms. We call them SO terms for sequence ontology. And they describe the consequence of a variant. Obviously, a variant can have more than one consequence, because a variant can have one consequence for one gene and a different consequence for another gene if those two genes fall in a close region, close to each other, where the variant falls as well. So each of the consequences are described in our documentation. You can see there's a link at the bottom. But you can see here that all of the consequences are color coordinated. So the reds and the more danger colors, the oranges and the yellows, these are variants and consequences that are going to have much more of a direct effect on the protein structure and function. So they might be things like the loss or gain of a stop or start codon, or a missense variant, for example. Here, these are the non-synonymous variants, which are yellow. The safer colors, like the greens and the blues, these are variants that have a less direct effect on the protein structure and function. So they might be UTR variants, or upstream and downstream variants, for example, or synonymous coding variants, which are green, for example. So when we're thinking now about missense variants, these are the non-synonymous variants. We can also think about them in terms of their pathogenicity. So we have two separate algorithms that we use in Ensembl to predict how detrimental a missense variant is going to be on the protein structure and function, and they're called SIFT and polyphen. So both of these algorithms, they take different pieces of information into consideration. So they look at the chemical change of the amino acid from the reference to the alternate, and they see how different the amino acid is. So whether you're changing charges or losing hydrophobic groups or something like that. And they also look at how well conserved the amino acid is, so whether it's at an active site, whether it forms important bonds within the protein or into other proteins as well. So these two algorithms independently both take similar information, and they predict how pathogenic a missense variant is going to be on the protein structure and function. So rather confusingly, they both work on a scale of 0 to 1, but they're inverse of each other. So for example, a SIFT score of 0 is deleterious, whereas 1 is more tolerated. And for polyphen, the inverse is true. So 1 is more damaging, and a score of 0 on polyphen is more benign. These scores in Ensembl, you'll be able to see them in the demonstration, but they're always accompanied with a description and the color coordination as well. So you're always, if you can never remember which way round the 0 to 1 is, scale for SIFT and polyphen, you can always just look at the description and the color coordination. So in Ensembl, we get variation data for a number of species. You can see all of the species highlighted here have variation data. I imagine you guys, having spoken to you a bit before, you're most interested in human, and then perhaps some of the other model organisms as well, like mouse and rats and zebrafish. And these species all have variation data. There's also obviously variation data for many species in the Ensembl Genomes project, which includes plants, fungi, protists, and metazoa as well. So as you probably well know, Ensembl doesn't perform any of its own sequencing experiments. We rely on people who identify variants to submit them to publicly accessible databases, and then Ensembl draws in data from those databases, and we take as much information as we can from these publicly accessible databases. So some of those examples, some of the major databases that we use and take data from is dbSNP, which is based in the NCBI. We also use the GWAS catalog, DGVA, which has information about large-scale structural variants, and then we also include data from OMIM, which is a database that has information about phenotype associations and OrphaNet and things like that as well. So we have a full list of variation sources, again, in the documentation that's linked at the bottom there. So there are two projects, and also probably a third that I should mention, that we also draw data from that we can display and look at in the Ensembl. So the first was the HapMap project. This is quite old now. It's been the data's been available for many years, and it looked at genotyping just over 1,000 different individuals from 11 populations from around the world. And we can get this data through Ensembl. So I know that it's currently now unavailable through the HapMap project pages. I think that's quite a new development. But that data you can still access through Ensembl, and I'm going to show you how to access that. But since the HapMap project, there was also the 1,000 Genomes project. So this was a more large-scale project that actually aimed at sequencing over 2,500 different individuals from many more different populations around the world. So while many of the populations from the HapMap project are also represented in the 1,000 Genomes project, there are many more different populations and also more individuals within each of those populations. So we get a better understanding of the variation data, of the variants that exist within the populations and how frequently the alleles are observed in the different individuals. The other project that I don't have a slide for that we've recently included data for in Ensembl is Nomad. So this is a project that is aimed at, it used to be called the EXAC project, so it's the Exome Aggregation Consortia. And it's now expanded its horizons, and it is aimed at compiling variation data from many, many different individuals, many different populations from around the world. So we also have the Nomad data as well that I can show you as well. So this is just an interesting and quite important concept to get our head around just before we start the demonstration. So this is how we refer to the reference and the alternate alleles in Ensembl. So if you attended the last webinar, you would have seen this example of a chromosome that I had that's made up of these contigs. So these blue blocks are the contigs that make up the human genome assembly. And this lady donated her genetic material to the Human Genome Project, and her genome sequence is used in this contig. So here you can see she has a T at this position that's highlighted in red. But in this hypothetical situation, this lady isn't actually very representative of the human population in general. So there's actually only 5% of people that have a T at this position. In fact, 95% of people have a G. And G, when we perform some alignments to the other primates, is the allele that we actually observe there as well. So G is what we call the ancestral allele. So it's where we observe what we observe in the other primates. And the T allele that this woman has here is also linked to some disease susceptibility as well. However, T is the allele in the config used, and she is the lady that donated her genetic material to the reference genome assembly. So T is the reference allele, and G is the alternate allele. So no matter the frequency, no matter the ancestral allele, no matter the disease association, the allele that's observed in the reference genome sequence is the reference allele, and any others are the alternate allele. So in Ensembl, we would represent that as T/G, so it's always reference/alternate. This is slightly complicated when we think about features which can be associated with a particular strand. So if we take our example from the previous slide and we look at the forward strand, so the lady had a T, which had an alternate allele observed as G. On the reverse strand, this is obviously A/C. And if we're thinking about a gene that falls on the reverse strand in this region, the exon sequence is actually either going to be A/C. So we're thinking about the reverse strands now. So when you look in Ensembl, the alleles in the database are always referred to as T/G. So you're always going to be thinking about the forward strand when you're retrieving data from the Ensembl databases. But when you're looking perhaps at variants in relation to genes, I'm going to show you an example of this, of a gene on the reverse strand with variation data. We're going to be listing that as A/C. So we're going to see an example. You're going to have the forward strand alleles listed, and then you're going to have transcript alleles described for the gene on the reverse strand. Some different databases can be different as well. We know that sometimes literature, people when they publish data can give sometimes more or less different information about the alleles that they've observed, and they sometimes don't specify the forward or the reverse strand. So just be careful about which database you're using and what you're reading when you're looking through the literature. Okay, so now I'm going to jump straight into the demonstration. While I load up that, I'm going to give you a chance to ask any questions if there's anything that's come up from the presentation so far. Okay, so I can't hear anything, so that either means that it's not working or that there's no questions. So I'm going to say that there's no questions, and we're going to go straight into the demonstration. So I'm going to jump straight to the Ensembl homepage. So we go to ensembl.org. And that will take you to the Ensembl homepage, which hopefully you are perhaps quite familiar with now. So what we're going to do is, I'm not going to describe everything on this page again because it's something that we covered before. And if you haven't attended the previous lecture, then I think Jenny will be able to share a recording with you as well anyway. So we're just going to search straight away for a gene of interest. So we're going to use the blue search bar, and this time we're going to search for the MCM6 gene. So MCM6. And then you can click Go. And then we can go to the Gene tab. So we click on the link at the top, and we're now in the Gene tab for the human MCM6 gene. So there's a number of places that we can already begin to think about variation data in relation to a specific gene. So I showed you this last time, but I want to quickly highlight some of this to you again now. So if you click on Sequence in the menu on the left-hand side, you can see that we have the sequence of the genomic region that surrounds the MCM6 gene. And we can actually add variation data to be annotated on this sequence by using the Configure This Page option. So if you click on Configure This Page, which is this blue button underneath the menu on the left-hand side, you can see that you have a number of different options to annotate and configure the display. And one of them is to show variants. So by default, we don't load variants onto this view. But you can click yes, in fact, or even yes and show links. And then you can save and close the window. and then your variation data will be loaded straight onto the sequence. Sometimes this takes a minute, so I'm just going to let that load.
Ben, this is Jenny. Can you please just remind us if you had loaded show variant with links, what does it link out to?
Okay, so I can just change this straight away. So if I click yes and show links, what we're going to actually see is the sequence is going to be annotated with variants. So each position is going to be color coordinated, and the letter of the base is going to be changed to an IUPAC ambiguity code. And you can hover and click on the variants on the sequence, but then for each line there's also going to be a list of links that link out to the variant tab for each variant. So if we had, for example, 5 positions in this line here where we had observed a variant, there'd be 5 links here in the side that would correspond with the variants that are loaded there. So hopefully this doesn't take too long. I don't want to waste time. Okay, so it's beginning to load now.
So while this is loading, I actually had another question. So you said the Ensembl incorporates polyphen and SIFT when looking at the impact of a missense change, for example, on a protein. Are there any plans to include any other predictors of effect, or, I mean, I know those, sorry, go ahead.
No, so we do have, in the VEP tool, there are other algorithms that predict effect, for example, on splicing and those sorts of things. And there, you can choose those parameters in your VEP query. I'm not sure is the answer. I'd have to talk with the people in the team that specifically curate the variation data and see. At the moment, I don't think that we exclusively use SIFT and polyphen. I don't think that there are any plans to include those sorts of predictive algorithms or any other types of predictive algorithms. Is there one that you had specifically in mind?
No, this has been a topic of conversation for us, just thinking about, looking at the impact of variants. And so one that we had been talking about recently was Revel, which I guess incorporates information from a lot of different algorithms. And it's part of our variant creation interface for ClinGen. And there are a bunch of others that we use, but I was just curious. I know that none of these are perfect, and
what was it called? Revel?
Curious. Yeah, REVEL.
So I think I've heard of this, and it does. I think it, like you're right in saying, it includes SIFT and polyphen, but then some other calculations as well. So
yeah, I believe so.
I'm not. So what I'll do is I'll get back in contact with the variation queue, and I'll see whether what their current status is on looking at including those sorts of algorithms. But I can get back to you perhaps more personally by email, and then you can update, because I don't know the answer at the moment without talking to some of my colleagues.
That's
okay?
Great. Thanks. Yeah, that's perfect. Okay. Thanks. I was just curious. Thank you.
That's okay. So now that the variation markup has actually loaded onto our sequence now, we can begin to see the variants. So as I mentioned, each of the positions where we have observed the variant is now color coordinated according to the sequence ontology consequences. So we can see, for example, in this region here, all of the variants are colored blue, and this is intronic. So we know that this is an intronic part of the MCM6 gene, whereas each highlighted space of sequence is one of the exons. So we can begin to see the light turquoisey blue is a UTR variant. So you can see this is a UTR region of this exon. And then we have the green and the yellow, which are the missense. The yellow are the missense and the green are the synonymous variants. So what we can do is we can actually find out more information about these variants by hovering over them. So you can see here, if you hover and click, you'll open up a pop-up window that has information about that variant specifically. So What you'll notice now is that each of the letters are not A, C, T, or G. They are, in fact, the RUPAC ambiguity code, so they describe the type of base change that we have. So rather a purine to a purine or a purine to a pyrimidine, the letter that is there describes the type of base substitution that we're observing at this position. So in this case, we've got a G to a C, which is a missense variant. So we're changing the amino acid that's encoded. This variant is obviously encoding a particular amino acid, and when we change it to a C, there's a different amino acid here. So what you can then see is along here we have the links to each of these different variants. So I don't think this is particularly useful, this view with the links added, because if you've got lots of variants in a single row, then you can see that you have lots of links and it's hard to really match them up. So what I prefer to do is to click on them individually, and then you can use the pop-up window and click Explore this variant. So that will take you to the Variant tab, which is what we're going to look at in a minute, where you can explore more information about that variant. Okay. So the next thing that I wanted to show you on the Gene tab is the phenotype association. So if you click on Phenotype, you'll be able to see the different diseases, traits, and phenotypes that are associated with this gene. So obviously we know that phenotypes and diseases, for example, can be associated with both genes and variants of the gene. So we have three different tables here. So the top table shows us the different phenotypes and diseases that are associated with the gene. So this is for the MCM6 gene, is associated with lactose intolerance, and this is an association that has been made by MIM Morbid. So this is this database that has phenotype association. So you can click directly on this link and it will take you out to the MIM Morbid database, and you can find out information about the disease association as well. That's going to have more background information about the disease and the association. The second table down is looking at the phenotypes and diseases associated with variants of the gene. So you can see all of the different variants. So you've got 5 different variants that are associated with 3 different phenotypes. So this is all variants that are associated with a phenotype, and there's 3 different phenotypes. We have body mass index. So one of the variants is associated with body mass index by the GWAS catalog, and the same for hip circumference. But in fact, all 5 of those variants have been associated with lactase persistence, which is the same as lactose intolerance. Obviously, it's just the opposite way of describing it. And this, again, has been associated by OMIM and ClinVar. So these are the 2 databases that have made these associations with the disease. If we scroll down the final table, it's just looking at orthologs of the MCM6 gene. So we're looking at mouse, for example, and rat orthologs, where obviously studies have been performed in model organisms and looked at the phenotypes associated with the gene as well. So we can see these 2 separate databases, MGI for mouse and RGD, have associated MCM6 with these 2 phenotypes as well. Okay, so if you want to find out more information about a particular the next thing I wanted to show you was the variants within the gene. So we've already looked at the variants in the sequence tab, but we can also view them in more tabular form. So if you click on variant table in the menu on the left-hand side, if you have any filters selected here, you can just remove them. So I think I had that from when I was playing with Ensembl before. So you can just remove all the filters from this top list here, and it will mean that you view all of the variants that are associated with the MCM6 gene. So these fall within a particular region. So what we know is that the downstream and the upstream variants are defined by the sequence ontologies within 5 kb of the gene. So what we know is that all of these variants that are given a downstream gene variant consequence, they fall within 5 kilobases of the MCM6 gene. So we can begin to find out some information about the variants. So they all have their own IDs, and these are hyperlinks, so we can find out more information about them, which is what we're going to do in a minute. You find their location, their alleles. So obviously we're looking at just that. So this is now the alleles according to the forward strand. So if you're not sure what any of these are, you can hover over the title and find out what we're going to be showing you in the different columns. So you can see this is an A as a reference allele and a G as an alternate. If there is population frequency, then you will display that as well. So you can see, for example, these 3 variants have been identified in the 1000 Genomes project, which you can see from this 1K flag that we have here in the table. So they all have population frequency data associated here. So this is the global minor allele frequency. And then you've got the consequences as well. So what you can do is filter this table. So if you want to filter by consequence, you can just click on consequences and turn them all off, and then turn back on Missense variant, for example, and click apply. So now I'm going to filter the table to only view the Missense variants. So obviously if I'd have scrolled down, I'd have been able to see them, but now I've just filtered the table. I'm just looking at the Missense variants. So again, you can see the alleles, you can see the source of the data. So dbSNP is one of the major databases we draw variation data from. They're all Missense variants. And because they're Missense variants, we also have information about the amino acid change that's involved here. So in this case, we've got a glutamate to a lysine at a particular amino acid position in the protein of interest. And then we have the SIFT and the polyphen scores as well here. So we've got the red or the green associated with the scores. Okay, so you can obviously export this table using the Excel button here as well. So you can filter your table as you like and then export it to Excel for your own further analysis and import into other pipelines as well. Okay, the last thing that I want to show you on this gene tab is the structural variants. So if you click on structural variants in the menu on the left-hand side, that link will take you to a page that describes and shows you the large-scale sequence variants that are associated in the region surrounding the MCM6 gene. So we have 2 separate tracks for this. We have the smaller variants. So obviously, these are smaller large-scale variants and then larger large-scale variants. So if you look at the legend at the bottom, you can see that purple represents a copy number variant. So we have that here and here. And then the red is either a loss or a deletion. So you can see that here as well. We've got different variants. And you can click on them, as we did before, and find out more information about the variant as well. Again, the page is just taking a little while to load. I don't think I need to particularly wait for this pop-up window to load. So all you'd find is information about the structural variant, and you would see the study that it was identified in as well. So you'd be able to link through PubMed to the study that is associated with that particular large-scale deletion. Okay, so now we're just going to quickly change direction. So at the moment, we've all been exploring variation data through the variants tab. So what we could have done, as I've pointed out at different points during the demonstration, is that we could have clicked on a single variant, either from the variant table or from the sequence view, for example, and navigated to the variant tab. So what I want to do now is pick on a single variant and go to the variant tab for that. So what I'm going to do is I'm just going to search for a variant ID that I know has quite good data and is quite interesting. So I'm going to do that now. Oh, I'm not sure what's happened there. Okay. Okay, so I'm not quite sure. So I'm just going to open up. Okay, so here is where we were before. Sorry about that. So I'm going to search for a variant ID that I know that has interesting data. So I'm going to use the search bar in the top right-hand corner of the blue bar at the top, and the ID is RS4988235. And then I can click enter. And again, my search will find all of the Ensembl features that have this ID. So we're just looking at this single human variant. So if we click on that link, will take us to the variants tab. Okay, so this is a tab that we haven't explored before during the webinar. So what you can see again is that we have a menu on the left-hand side that's going to display specific information about the variant of interest. So at the moment we're in the Explore this variant option. So this just contains a basic summary of our variant of interest. So this is an intronic variant. Its alleles are G/A/C. So the G is the reference allele, but A and C have also been reported at this region as well. So the ancestral allele is the G. So in fact, when we perform our alignments with other primates, it's a G at this position. And then the minor allele frequency, we actually observe an A is 16% of the global population. You can find out other information about the variant as well, like its location. You can find out some information about its HGVS nomenclature, which I think maybe you might be interested in using in your analyses. HGVS obviously indicates perhaps sometimes the protein and transcript effects as well. So you can use these and show, for example, the HGVS nomenclature. So each of these links at the bottom are actually the same as the links in the menu on the left-hand side. So, for example, this genes and regulation link here is the same as the genes and regulation link in the menu on the left-hand side. So what I'm going to do now is just explore some of these options in the menu on the left-hand side. So first I'm going to click genes and regulation. And I'm going to hide this HGVS nomenclature as well. So we always get this summary information at the top, so I'm going to hide that just to simplify our view so that we're not getting confused. And then when I scroll down, we're going to be able to see the genes and regulation. So this is a page that displays the consequences for our variant. So as I mentioned, a variant can have more than one consequence. So each, the variant has its consequence analyzed for all of the transcripts of the MCM6 gene. So what we can see here is that this variant only affects the MCM6 gene. All of the different consequences are relating to this MCM6 gene here. But each of the three transcripts, so we have the one ending in 156, the one ending in 902, and the one ending in 209, have all been analyzed for their consequences. And in fact, each of the different alleles, the A and the C, each of the alternate alleles have been analyzed for their consequence in relation to each of the transcripts. So for the three transcripts of the MCM6 gene and the two alternate alleles gives us the 6 lines of this table. So what you can see here is that the allele has been listed here as the A or the C, which is what we had here, the A or the C. But because the MCM6 gene is on the reverse strand, we have the transcript allele listed as the T. So this is what I was referring to in the presentation, where if you're thinking about a feature on the reverse strand, then you obviously have to also think about whether the exon sequence of the reverse stranded feature is going to be changed. So this is obviously the transcript allele is a T in this case. And in this case, when we're looking at this transcript with this variant alternate allele, it's an intronic variant. Underneath, you can find information about gene expression correlation, so you can see how this variant affects the expression of different genes in the region. So this is data taken from the GTEx EQTL project, so you can explore that. And you can also see whether the variant affects any regulatory features as well. In this case, there are no regulatory features that overlap with our variant of interest. The next thing I want to show you is the population genetics. So if I click on population genetics on the left-hand side, we're now going to be able to see the population frequencies, in this case from the 1000 Genomes project, for this variant. So if there's data in the NOMAD project or the HapMap project, you would also be able to find that on this page as well. So what we can see here is firstly these pie charts at the top. So these indicate the prevalence of the different alleles in the different populations of the 1000 Genomes project. So here we can see all. So these are the global, all of the individuals from the 1000 Genomes projects are represented in this table. And so 85% of individuals in the 1000 Genomes project, we observed a G, and in 16% we observed an A. So then we can split it down by what we call superpopulations. So here we're looking at all of the African individuals, the American individuals, the East Asian individuals, the European individuals, and the South Asian individuals. So as we can begin to see, while as a global percentage we observe A at 16%, this is actually at 50% in Europeans and at 0% in East Asians. So this is a really interesting example because it actually, as we are going to see, is involved and is implicated and associated with lactase persistence. So the fact that it's much more prevalent in Europeans than East Asians is actually indicative of how dairy farming spread around the world. And I think it's quite a neat example of how we can use genetics to understand human evolution and history as well. So I think that's quite a neat example. Underneath there, there's a tab. Oh, so the last thing that I wanted to show you how to do is to then interrogate these superpopulations even further. So what I've done is I've clicked on this button here, subpopulation. So this is the table, this is the view that you should have by default at the moment. But if you want to investigate the European populations, for example, in more detail, you can click on the subpopulations option underneath the pie chart, and it will open up a whole new set of pie charts that each describe the allele frequencies in the different populations of the European superpopulation of the 1000 Genomes Project. So again, you can see how the G and the A is distributed across the different populations. Underneath that, we have a tabular representation of exactly the same data. So you can see here we have the G and the A. So we have the G at 84% and the A at 16%. So these are the allele frequencies that are described here. And then in brackets, we have the raw numbers of individuals from the 1000 Genomes Project. And then on the right-hand side of the table, we have the genotype frequency. So obviously, we know that each individual has 2 copies of each chromosome. So in fact, the homozygotes for G were observed at 75% of individuals. Homozygotes for A was at 8%, and the heterozygotes AG were in 16% of the global population. Again, you can split that down by the different populations of the different 1000 Genomes Project. You can jump to the different projects. So again, for example, you can jump to the HapMap project or the UK 10K project or the Nomad project, and that will jump you down the page to find out information about the allele frequencies that are observed in these different projects. Okay, so the last thing that I wanted to show you on the page is the phenotype data. So if I click on phenotype, this is going to take you to a page that shows you the different phenotypes that are associated with, this time, the variant of interest. So you can see, for example, that it's Bosdi mass index, as we saw before, and hip circumference. So this is something that we saw on the gene tab in the tables on the phenotype option. But again, we have lactase persistence, which was annotated by both ClinVar and OMIM. So if we want to find out more information about this phenotype specifically, we can click actually on the name of the phenotype. And that's going to take us to what we call the phenotype tab. So if I again just remove the filter, like I did before, we're going to be shown now a table that gives us all of the different features within Ensembl that are associated with this particular phenotype. So in this case, lactase persistence. So here you can see all of the, I think, 10 variants here that are associated with lactase persistence in Ensembl. In fact, so some of these are duplicated because they've come from both OMIM and ClinVar. So you can see here, this is a table. Obviously, if you were looking at something like obesity or blood pressure, this table would be many, many hundreds of rows long with each variant and gene associated with the phenotype listed in the table. Okay, so that brings me to the very end of the demonstration for the variation data. I just wanted to quickly now jump into the VEP demonstration. So hopefully this is going to be very quick. I'm not going to be able to show you everything, but hopefully I'm going to give you a quick flavor of what you can do with the VEP. So just as a quick overview, you may have done your own sequencing experiments or identified some variants of interest, for example, with a genotype array, and identified a SNP of interest, and you want to find out about what it does and its functional consequences. So this is why we've developed a tool called the Variant Effect Predictor. So the VEP, which we name it for short, allows you to input your own variation data that you might have found in the literature or generated yourself, and find out for those variants the effects on the genes, the transcripts and the proteins, so you can retrieve the consequence terms, and you can also retrieve pathogenicity scores. If the variant has already been described in Ensembl, then you can get the frequency data and the literature citations, for example, and you can also get the regulatory and splicing consequences as well. So in Ensembl, we have a number of species that have variation data, but in fact, every species in Ensembl you can use with the VEP. So as long as there's a reference genome assembly, and obviously your own variation data, your own variation data is going to be analyzed in the context of the reference genome assembly. So any species that you're studying in Ensembl, you have variation data for, you can use with the VEP. And even if you're studying a species not in Ensembl, you can even set up your own cache using a gene annotation file and FASTA files. You can set up your own cache and use the VEP to run your variation data against your species of interest. So even if you don't have a reference genome assembly in Ensembl, you can set up a cache and analyze your own variation data there as well. So we can use the VEP in two major formats. You can also use the REST API as well, but we like to think of the web interface as being the most user-friendly option, so it doesn't require any computer programming knowledge or script writing. So this is what I'm going to quickly demonstrate to you now. And then we also have a standalone Perl script as well. So this is a Perl script that you would run from the command line, and it's more flexible, much more quick, and you can analyze many millions of variants in a matter of minutes. So I'm just going to skip this slide. It's just to let you know that if you're using the VEP standalone script, you can use plugins and add extra functionality to your VEP job. So in the course book, we're very, very quickly going to look at the effect of 4 separate variants that we've described here using the VEP. So I'm just going to jump out of the presentation again, and I'm going to copy my variation data here. This might not be important for you to do now, but I just want to show you how to use the VEP. So I'm going to again jump to Ensembl. And as you can see, we have a link for the variant effect predictor on the home page. So you can click just straight here, and you can see here's all the documentation for either using the web interface, the standalone script, or the REST API with the VEP. But if you want to just launch the web interface, you can click here. And if you've submitted VEP jobs before, then you'll have a list of your previously submitted VEP jobs here. If you've never submitted one before, you'll have this page. So I'm going to click New Job, and this is the page that you should be presented with if you haven't run a VEP job before. So this is the submission form that you need to fill in. So the first thing is to select your species of interest, human, and then to give your VEP job a name. So I'm just going to call mine example. And then you need to input your variation data. So you can input your variation data in Ensembl default format, which is what we're going to use, or you can use VCF formats. You can use variant identifiers, like the RS numbers that we were looking at before if you've searched literature. And you can also use HGBS notation as well. So we can paste our data directly into the text box. Oh, I think I didn't copy my data from the presentation. Okay, so this hasn't worked for me. So what I'm in fact going to do is I'm just going to use, you can populate this text box with some example data in each of these different formats. So I'm just going to click on Ensembl default, and instead of using the example data from the presentation, we're just going to look at this sample data that we've populated the text box with by clicking on this Ensembl default example. So this is normally just meant to be used to show you how the data is meant to be formatted in these different cases. So you can click on the different formats and it will automatically populate the text box with your data of your format of interest. So we're actually going to use the example data because we couldn't get it to work from the presentation. You can also upload a file or you can provide a URL that is directed to a public directory that contains a much larger file if you've generated a large file with many millions of variants, for example. Then you need to select some of the different parameters that I just want to very, very quickly highlight to you. So you can choose, for example, to see the genes that are affected. You can choose to find the co-located variants. So you can see if the variants already exist in the Ensembl database, and if so, you can retrieve their population frequencies from a number of different projects, like the 1000 Genomes project or the Nomad project. You can get the literature citations. You can get the pathogenicity scores and predictions. So you're going to get the SIFT and polyphen score and predictions as well for any missense variants. And you can also get any regulatory region consequences as well. And finally, at the bottom, you can choose to filter. So there's some filter options as well. Once you've selected all the parameters you want, you can click Run. So I'm just going very quickly because I can appreciate that you probably need to finish by 1 o'clock. So what happens is that your job is added to the queue here, and the page automatically refreshes every 10 seconds. So I'm going to click on this previous job I've done, and hopefully we'll be able to see the data here. So this is an example of the results that you would get. So you can see here a summary at the top. We've processed 6 variants in this example, and you can begin to see, so we've processed 6 variants, and you can begin to see the breakdown of information for the variants at the bottom as well. So you've got the location and the allele, which would have been described in the input data. We've got the predicted consequence. We've got the affected gene. So again, one variant might have multiple effects. So a single variant can have multiple rows in this table. We've got the affected genes and transcripts. And you can also scroll across and find the SIFT and the polyphen scores, for example, where there are missense variants. In this case, we don't have any. And also the population frequencies as well, for example. Okay. So I know that was very, very quick. I apologize for it. What I'm going to do is actually point you to a much more comprehensive BEP tutorial as well at the very end. So I just want to quickly just present 2 or 3 slides before I finish. So as Jenny may have mentioned to you before, I've organized a feedback survey that I would really appreciate it if you filled out. So I've been organizing this webinar series with Jenny, and I would really like to know whether you found it useful, whether there's anything that you prefer that we would have covered, or if there's anything that you would like to have covered in the future, and just to let you, so you can let me know whether you found my presentation style interesting and engaging and those sorts of things. So you can actually find a link to the presentation on the training pages. I think Jenny's going to send around the link as well. So just to think about the different data, we had the 4 different types of data that we can access with Ensembl. So in our 2 webinars, we've looked at the gene annotation and the variation data. We haven't had time to really explore the regulation data and the comparative genomics that we also have available in Ensembl as well. So all of this data is organized and available through the web interface. So this is how we've been mainly exploring Ensembl in the 2 webinars. But you can also access the data through Biomart, through the Ensembl databases by querying them with the APIs or directly with MySQL. Or you can download FTP files. You can download files from the FTP site that contain genome-wide data for each of the species in Ensembl as well. So if you do want to learn more about Ensembl, you can definitely do it in a number of different ways. So we have a number of different online courses that are available through the EBI pages. So the EBI is the larger organization that Ensembl is part of. So you can find some Ensembl tutorials on their training pages. We also have a YouTube channel called Ensembl Help Desk, where you can see lots of recorded demonstrations about how to use Ensembl and some of the tools. So this is where I would go if I were you, if you were interested in getting a more comprehensive tutorial about using the VEP, or perhaps looking at the regulation or the comparative genomics data that we haven't had a chance to cover as well. And then we also have a help desk as well. So if you want to contact us about anything that you find in Ensembl, or if you have any questions, or if you get stuck, or if you find any errors, and then please contact helpdesk@ensembl.org, and this will help. This will allow you to ask us any questions, and we'll get back to you trying to resolve them as quickly as possible. If you do use Ensembl, which it is a free resource, so we really hope that you find it useful and that you want to use it, then please just cite us. It really helps us understand who is using Ensembl and what for. So while it's free, we do appreciate everyone that cites Ensembl in their work as well. So we have a general paper published in NAR every year that you can cite. And if you want to keep up to date with Ensembl, you can follow us on our social media channels, but also through our blog. So this is where we publish important information about the release cycle, the new data that's being incorporated into Ensembl, and any maintenance downtime, for example. And then this is the whole team that make the resource possible. So this is all of the Ensembl and the Ensembl Genomes team together as well. And then that just leaves me to thank everyone. So I obviously have quite a nice job in that I get to present this wonderful resource to everyone, but it's actually everyone else's hard work that makes it so good. So this is everyone that you would have seen on the previous slide. And then we get lots of funding from a number of different bodies as well. So the Wellcome Trust provides us with our core grant, and then we also get important grants from other projects such as the BBSRC, NIH, Open Targets, and the EU and Ensembl as well. So I think it's about 1 minute to 1. So I think that was a very quick overview, but hopefully it was useful to you. I'm more than happy to stick around for a few more minutes if you have time, and if you have any questions as well. So thank you very much.
That was great. Thank you so much, Ben.
That's okay.
Does anyone have any quick questions for Ben before we leave the call? I know some people might have to leave now, but as Ben said, he can stick around for a couple of minutes.
Yeah, I appreciate that many people would have had to have gone. So if you do have any questions, then you can either ask them through Jenny, you can contact me, or you can contact the help desk as I described before as well.
Thanks so much. I do have a quick question actually, Ben, about the VEP. So it is giving you the effect of a variation on different transcripts, but I guess the effect on clinical phenotype is going to depend on the tissue in which that transcript is expressed, or how common that transcript is. So is that information incorporated into the VEP in any way?
No, so expression data is something, no, you're exactly right. So yeah, you are right in your, when you say that the effect of a variant might be dependent upon obviously tissue-specific expression of particular transcripts, but that's not something that we have in Ensembl. So in the last webinar, I mentioned that we had the gene expression patterns that we had from expression alleles. That's only gene-specific expression, so we don't have any information about transcript-specific expression in Ensembl, and therefore there's no way that the VEP can interrogate how a variant might affect transcript expression in particular tissues. That's not something that we have, I'm afraid.
Right. I guess I'm just thinking, like, if you had the impact on, like, say, 3 different transcripts, like, if one of those was, like, by far the most common, you might be more likely to think that this is the one that's really going to potentially impact the phenotype. So
yeah, I mean, so when you go to the VEP, there are a number of different options that you can select. So you can find out information about the transcript in terms of its transcript support level, so whether it's has a good level of annotation behind it. And you can also find out if the transcript is protein coding, so you can find its biotype. And you can also find out if the transcript is listed as a principal isoform. So these are some of the parameters and flags that we looked at in the last webinar. So the APRI annotation is something that I think is quite useful in this case, because it's a separate annotation by an independent group, and they've gone through the different transcripts of the genes and annotated which transcript they think is the principal isoform. And so when you go to the VEP, you can click an option that will tell you if the transcript that's affected is the principal isoform. So you can see that, but obviously it's more based on like an algorithmic decision previously. Like, it's not based on, I mean, I'm guessing it is based on expression data, but there's something that's done before it even gets to Ensembl, if that makes sense.
Right. But knowing what's the principal isoform, yeah, that's really helpful. So yeah, that's great. That answered my question. Thank you.
Yeah, so that's an option that you can select in the VEP. I had to go through it so quickly because I realized that we were running out of time a little bit, I think.
No, this has been great. Thank you so much.